So today I'm going to tell you a little bit about some of the work that is going on in synthetic biology with a little bit more emphasis on our work, of course, in telling you how control theory is becoming more and more prominent in this field as we move forward, okay? So control theory has been historically applied to a bunch of different engineering systems, right? From mechanical, aerospace, right? Virtually every engineering systems that you can think of has feedback control inside, okay? And this is starting to become more and more present in synthetic biology as well, okay? So I hope by the end of the talk, right, you get away with a little bit of feeling what synthetic biology is about, right? What are the challenges in the field, right? And how control systems, right, is being used, right, in order to solve those challenges. So in any case, so I'll start with a little bit of history, okay, on synthetic biology. So I think right now everybody agrees that the birth of the field was in about 2000, okay, when people were able to put genetic engineering tools, such as cloning, okay, cutting and pasting together pieces of DNA, into an ability of creating small, what they call circuits, okay? The first two famous circuits that were built were an oscillator, right, where you have essentially three proteins that repress each other in a ring fashion that create oscillations, okay? And then a toggle switch, which is a system that can keep in memory two different states, okay? And there was a long era of the field, which I call the motifs era, where the purpose was to engineer simple systems to understand biology, right? So based on the fact that if I can engineer a system that works as predicted, I kind of know, right, how biology works, okay? So that's essentially how the field started. But then there has been a transition at some point that people realized, wait a second, we can actually put together all these simple systems to create sophisticated systems that are smart enough, okay, that can tackle a series of societal challenges, okay? And here I'm just giving a few examples, right, of efforts in the field. So the first one is really tackling cancer, okay? So what you can do right now is essentially you can program bacteria to essentially sense the presence of cancer cell, okay, and react to that. So they can essentially sense, right, actuate, in the sense that they lyse open and they release killing agents, okay, so that they go to the cancer cells and they kill them, okay? And this is really going on right now. Of course, this is still in mice, okay? But these are actually things that are happening right now. It's not fiction, okay? Another very exciting field, which I'll talk a little bit more in this presentation, is the control of self-fate, okay? So the ability of being able to reprogram, okay, the fate of a cell, for example, from a skin cell to a stem cell to a heart cell, okay? That biology has done an enormous amount of advances in that area, but the question, can we control, right, the process? And with synthetic biology, it turns out that the technology, right, to control the process is getting there. And of course, there are, just want to point out, more traditional applications, such as in biofuels, where these have been going probably on for 10 years, where you can put bacteria that are suitably programmed in an environment that can sense some sort of waste, right, and turn into some sort of biofuel, such as ethanol, okay? In any case, so what I'm going to focus on in this talk is essentially how do we make the transition, right? That's essentially where the field is at the foundational level. How do we make the transition from simple motifs to systems that they can sense, compute, and actuate and interact, right, with endogenous cells? So in order to do that, I will just give you a glimpse of this example that we come back later in the talk, which is really reprogramming self-fate, okay? So the notion of reprogramming self-fate goes back now to about 10 years. So initially, people were thinking that just like a ball rolling down a hilly landscape, the process of self-fate specification is unidirectional, okay? So you can view these top of the hill, like the pluripotent stem cell is this cell that has the ability to differentiate in essentially almost all cell types, right, in your body. And then as you come down the hill, right, you have more and more specified cells, right, at the point that you can have skin cells or other sorts of blood cells and so on. So if you move this way, the differentiation potential increases, okay? And this feature has been used since probably the 40s, okay, to view the notion of cell differentiation developed as unidirectional. But the idea is that now we know, at least from 10 years ago, that you can actually force, right, transition in between cell lineages, and you can also go all the way back up to the pluripotent state, okay? How is this done? Okay, so we'll come back to the details later, but essentially, the way you do, okay, so I'll focus specifically on this process of induced pluripotent cell reprogramming, okay, and by the way, these folks got the Nobel Prize, right, in 2013, right, for discovering this. So essentially, here is the traditional way in which this is done. Okay, you have this complex circuitry of the cell that actually creates this landscape, right, this energetic landscape with interactions between genes and proteins. And then what you have artificial overexpression of specific factors, okay, that are supposed to push the cell to your desired phenotype, okay? So, from a control system point of view, you can view these as the system to be controlled, right, it's the cell. The output to be controlled, right, of interest is really the type of the cell, right? And the input that you can control is essentially how much you are introducing artificially of these specific factors, okay? If you view it like this can be reviewed as an open loop control strategy, okay? So open loop, it means that you are applying, right, an input, and you are hoping that with that specific input, the output is going to reach exactly, right, the point you want, okay? It's as if you're driving a car in a hilly landscape, you can only apply acceleration once, and I'm telling you, I want you to stop exactly in 50 yards from where you are, right? Pretty hard task. And in fact, it's not surprising that the efficiency of this process is extremely low. You have success rate of about 1 cell over 10,000, okay? And the process is also very slow. It takes many, many weeks, okay? So, what can we do with synthetic biology? Essentially, the idea, right, you can transform an open loop strategy into a closed loop strategy, where in principle you can measure, right, your output of interest, which is the cell phenotype. You can process that through a smart genetic circuit that is built to steer, right, the cell through suitable transcription factor concentration all the way to the phenotype you want, okay? So essentially we go from a paradigm where you are not measuring the output of your interest to one in which you measure it and you adjust the input based on that, okay? Of course, the expected outcome here is that you hope, right, to increase the efficiency, right, makes the process faster, right, and less expensive. But anyway, we'll come back to that later. What's the technology, right, that goes in here, okay, that would allow us eventually, one day, right, to do this? So, here is just a little bit of background of how we build biological circuits, right, in synthetic biology. You start from a library of parts, okay? For us, parts are promoters, genes, right, that express your proteins. Proteins is the signal that you have in the cell that control all important behaviors of the cell. And then you combine these together to form modules, right, that we'll talk about. And then the idea is that you combine these modules into bigger systems, okay? For example, this is a classifier. So it's a system that's recognizing a specific chemical signature that you have in the environment by looking at the combination of small molecules that are present, and then gives an output saying, okay, if the signature is correct, the output fluoresces in green, for example, right? So it's a classifier. And then the idea is that you want to put all this into a cell so that the cell can sense, compute, and actuate exactly the way you want. So this is a very old movie, right? And it's a neutrophil, it's a white blood cell that is chasing a bacterium in the bloodstream. Now imagine that you were able to re-engineer the sensing, the computation, and the actuation ability of this cell, so that is actually chasing a cancer cell, okay? So that will be pretty amazing, right? Well, it turns out that we are, right, doing a lot of efforts, right, to get to that point. So, but where are we? So, you would think that in a mechanism where you proceed bottom up, right, you have parts, you assemble in module, you assemble in circuits, and you put in the cell, the idea of modular composition is crucial, right? So that's essentially the idea that if I have a system, right, like, I don't know, an amplifier, for example, I connect it with something else, it's still keeping its property unchanged, right? Think you combine it with something else, and all of a sudden an amplifier is decreasing the amplitude of the signal, right? So, of course, things wouldn't work anyway. So, Unfortunately, modularity is not a natural property of this system. Okay, so the reality today, which is really a big point where people are doing a lot of efforts, is really that whenever you have designed a lot of really cool small circuits, once you start putting them together in the cell, they change their function. And it's often really hard to predict how that function is changed. Think about it. Think about having, your computer. You are essentially at some point implementing a program, and your computer doesn't like it too much, and the computer turns into a TV, right? Or something different. So that's really a case, right, where essentially what you are implementing, right, in the cell is changing the behavior of all the circuitry around it. Okay? So that's a big problem that in the field is called context dependence. So it's the fact that the behavior of any module you have, unfortunately, depends to some extent on the context around it. So in this talk, I will first focus on this transition, okay? So what are some efforts, right, that we can make in order to go from modules, right, to systems? So, and I will start with two more specific examples of what can go wrong, so we can fix the ideas, okay? So here I'm taking a zoom at this circuit that is composed of sensors. We have a clock that periodically samples the environment, and then we have an AND gate, and then an amplifier and the output, okay? So let's look, for example, at the interaction between a clock and its downstream systems, right? In electronics, if you have a clock that is keeping the time, right, and you connect it to downstream circuits, the period is not changing, the clock is still functioning, right? In this case, things are a little bit different. If you take a closer look at the clock, in which you have these two genes, okay, one is activating, one is repressing, this is self-activating. By the way, for the people that know a little bit about clocks in electronics, this is a hysteresis-based oscillator. Then what you have is that, okay, let's take the clock and let's connect the output protein to a downstream system. Ideally, I would like to view this protein as transmitting the signal downstream. Unfortunately, that's not quite what happens, because when this protein gets transmitted to downstream, it's not taking part in the reactions. So we are talking about chemical reactions, okay, that are seeing a decrease in protein A here, and as a consequence, your clock doesn't work anymore, okay? So that's an example. You connect a clock as a signal generator, you connect it to something downstream, right, and the signal gets destroyed. So this is an example of retroactivity, okay, which is a notion that Eduardo and I introduced in 2008 to essentially capture the concept of loading, okay, in biological circuit. So this is essentially saying that direct connectivity to downstream systems, right, is changing the behavior, right, of the signal you're transmitting. And of course, we know in electronics, back in the days before operational amplifiers, we had similar issues, okay? But then operational amplifiers came into picture and actually solved a big deal of that problem, okay? So let's see now another problem that can happen, okay? So you have resources in the cell, okay? So the cell is providing all key enzymes. For the people that remember a little bit about the central dogma, right, in molecular biology, you have that the process of protein generation is taking resources. So these are RNA polymerase, ribosomes, right, that are necessary to go from DNA to protein, which is the signal, okay? Everybody is sharing the same resources, and the cell is producing a certain amount, doesn't have infinite resources. So what do you think? That if one part of the circuit gets more, another part gets less, okay? And this may harm circuit behavior. So here is an example. Let's look at this very simple cascade that you have here, okay? This is an activation cascade. It is virtually found in any genetic circuit, right? Typically serves to amplify a signal. So you have an input U that can be a protein that is activating another protein, that is activating another protein, and overall you would expect to have somewhat ultrasensitive behavior, but definitely positive, okay? Some years back, a student of mine was taking this cascade for doing something else with the idea of creating a bigger system. He went to the lab and built it, and this is what he found. And he came back to me almost crying and saying, look, I can't even build the simple circuit, right, that you can think of as simple cascade, right, it's done of two genes. Clearly, this is not an increasing behavior, right? These two are different, okay? So what's going on here? And of course, the students were really depressed at that point, saying my career is over. And then I said, no, there is something interesting here to understand, right? Every time, right, you make an experiment, right, and the outcome is different from what you predict, that's where the interesting research comes into picture, right? And in fact, it turns out that that was the case. In fact, if you think back, right, and now you put the system back into context, okay? The context here is what matters, right? So you have your RNA polymerase, your ribosomes, right, that are getting used by these production processes. So once you add your input that is supposed to produce this protein, these resources are demanded more in this node, okay? So less are left in the output node, which is what you observe, and if this arrow becomes really weak, actually, right, you see a decrease in behavior instead of an increase, okay? It's really, right, a competition problem that is created between two genes here that isn't really in your head supposed to be there, but it is, okay? Of course, in the case of infinite resources, this wouldn't be there. So, this is another example, right? Sharing limited resources, right, is actually creating very subtle couplings. And here is just showing RNP and ribosomes. But you can think of, especially in mammalian cells, which I'll get a little bit later in the talk, there are many other crucial resources, especially for transcription, right? Transcription is an extremely complicated process that has a lot of transcriptional cofactors. In principle, they can all be shared resources, okay? And that, I will say, is a big part where the field is trying to make a lot of efforts, right, to understand and actually get rid of these effects. So, here is an overview of the talk. I will touch two main points here about context dependence. So the first one is direct connectivity giving rise to loading, right? The example of your clock transmitting the signal gets loaded, drops down, right? You fail to transmit, okay? So the second issue is the issue of cellular resources, right? Now you have that this system supply a load, right, to the circuitry of the cell that is providing the resource. Of course, the cell is designed to produce so much, right, for the endogenous system, and you are perturbing it really brutally sometimes, okay? The worst case, the cell dies, right? And so what I will do then, I will go a little bit more in the application and saying, okay, if we can fix all this, okay? So here is a very intriguing application that we started looking at a couple of years ago, and I will finish with that. So let's look a little bit more at retroactivity. So this is an example, okay, of a genetic circuit. We have essentially a transcriptional component, right, with an input and an output here. And now we are asking the question of how the input-output response changes when you put some loads, okay? In the context of biology, right, or decentralized circuit, the load is typically a DNA binding site, because that's how connection between modules happen. Transcription factor binding to binding sites, okay? Here is a step response, okay? So you apply a step input, and what you see that when you have load, you have some slowdown of the response. Now, if you want to look at the frequency response, so you are applying a time-varying, right, input here, what you get is that the isolated system winning without the load starts to behave very differently from the way of the connected system behaves, and ultimately the signal gets lost, okay? So this is telling you, right, how we saw in the clock that the signal you are transmitting, it's failing, right, to get transmitted. So this is saying that retroactivity slows down the response of transcriptional component, reduces bandwidth, and this has been shown, of course, these are experiments in yeast, it has been shown in E. coli, it has been shown in mammalian cells, so that's a pretty, right, general effect. But this is a problem where I think also with the help of our collaboration with Eduardo, we actually made a lot of progress, okay? So what is the idea? This is really the idea that Eduardo and I inserted in 2008, okay? Which is the idea of insulation devices, okay? So what if you decouple the upstream system from the downstream system, okay? And put in between them a smart device, okay? Which we call insulation device, okay? So like in electronics, right, you put between an upstream system and a downstream system what's called an isolation amplifier, okay? You can do the same here, okay? So what is this insulation device doing? It must have two key properties, okay? It must essentially be very careful, right, in not applying a big load to its upstream system, okay? In electrical terms, right, it means it has very high input impedance, okay? And on the output, it must be able to keep the output response to you essentially independent of an arbitrarily big load imparted by downstream systems. So in electrical terms, it means its output impedance is really low, okay? So that's essentially the concept. So how do we address the engineering of this? Well, I'll focus on how to make the effect of this arrow on the U to Y response really small, okay? Because that's where feedback comes into picture, okay? So, and this is essentially the idea of high gain feedback that was used probably among the first times in a concrete application, right, in Black's amplifier, right, in the 20s, where people were trying to put amplifiers in series, right, to create the telephone line that will go from California all the way to, right, New England. And the problem was that these amplifiers had distortions, were very sensitive to temperature changes, right, and Black came and said, let's put a feedback loop around it, right, to fix the problem. So let's try to do the same, okay? So that's the basic concept of disturbance attenuation with feedback, okay? So that's what I teach in Feedback Control 101, right? So you have the input and the output, and you have your disturbance here that in our case is this loading. You can view that as a disturbance that you want to get rid of, okay? How do you do? Well, you take your output, you compare it to the input, and if you have an error here, if the error is positive, right, essentially you are going to amplify that and increase Y. If the error is negative, you're going to decrease Y, okay? So if Y is too high, you decrease it. If Y is too low, you increase it, okay? So that's the concept of feedback. This gain G is really high, okay? Essentially, you can solve this block diagram. You get essentially that Y is going to have two terms, one depending on the input, one depending on the disturbance. But if the gain G is really high, you can get rid of this term, okay? So that's the concept of disturbance attenuation, okay? Roughly speaking. So how do we implement this in a biological system? Turns out that it's useful to rewrite this block diagram this way, okay? So your input is getting amplified, and then your output has a large negative feedback. So what this is telling me that if I want to implement this biologically, I need two processes. One, to amplify the input a lot, so that by the time it gets compared to the disturbance is really large, right? But now you want to keep the output back down, right, to the original value, okay? And that's why you need an enhanced degradation on the output, okay, in biological terms, okay? So does this general concept make sense for now? Yeah? Kind of. So how do we do that? So here is when we go back to biology, okay? So at this point, right, we were talking to our biology collaborators and we were asking, okay, here amplification is key, right? Whether you want to amplify the degradation of your output or amplify your input, amplification is key. So what is the biological mechanism that gives you signal amplification? Okay? Does anyone have an idea? I heard something here. Signaling, right? So signal transduction, right, is really what's meant to give you amplification. And that's what we did, okay? So let's go back where we were, okay? So we essentially took our load driver to be a cascade of two signaling motifs, okay? So signals essentially are amplified through reactions that are called phosphorylations, okay? And you have a mass degradation to arrows that come back that are called dephosphorylation. Any disturbance that you add is going to be cancelled, okay? So you can write a very simple equation that is actually going to show you that, and you can look at this or our paper in 2008 with Eduardo, okay? So, of course, this was the original idea. We had a very simple biophysical model that demonstrates this concept, right? That this very fast cycling of phosphorylation makes that any disturbance is essentially cancelled. Of course, we wanted to do the experiment implemented, okay? But then when you go to your biology collaborator and tell you have this beautiful biophysical model is predicting this fantastic property, they're going to be very skeptical at you because they say, okay, if I build this, right, the biological system is so complex, they are biophysical model, right? What guarantees do I have that that's actually what's happening there? So and that's why it took us, this is still the time in which we weren't doing the experiments yet, but it took us about 3 years to convince this person to do the experiments. And we went from a model that had one state variable, I'm talking about ordinary differential equation, to a model that had 56 ordinary differential equations. Okay, we had both deterministic and stochastic models. We showed the result, and finally, our collaborator was convinced, yes, okay, let's build a system. And that's what we did. And that's actually why after that I said, okay, I should put up my own lab and believe more in my biophysical model, do the experiments, and so on. But I would also want to emphasize that we in engineering, we tend to write the simplest models first to get an idea of the concept. It's actually similar to what physicists do in some sense. But then I would argue, right, that biologists are also right, that there is a whole way of complexity in between your simple concept and what you implement. So at some point, you have also to do big scale computational models, right, that are somehow able to reperpetuate, right, your property. So in any way, so this was the original performance that we had without insulation device. So after 4 years of a BE student working in building the system, this was a system that was partially extracted from mice and partially from yeast and replaced in yeast cells, okay? We went from a performance like this to a performance like that, okay? So we had perfect disturbance attenuation, okay? And this is when the BE student that had actually trusted us, theoreticians, right, that came shocked. This is the first experiment that he did. He came shocked and said, theory works. And I said, yes, it does, but I now understand where it's coming from, right? In this big complexity, a simple concept, right, that comes, right, can actually be very powerful, right? But that can be surprising as well. So, anyways, with this, we actually were very happy because we essentially now think that we have a good grip, okay, at least how to solve this connectivity issue, right? You put in between an isolation amplifier, okay? And it turns out, I won't go into the details, that I show you one implementation through this cascade of 2 systems, okay? But it turns out that nature offers a plethora, okay, of signal transduction motifs. So your library, right, of potentially orthogonal devices that you can build is pretty large, okay? And we have a recent paper in 2017 that actually look at all this library, right? And what are the best structures that you can find in nature, okay? So anyways, so at this point, this was, I would say, 2014, okay? And we were super happy and said, okay, we got rid of the loading problem from direct connectivity. But this is when my depressed student came with this. And of course, right, at this point, we went back to this and say, okay, we solved one problem, but here comes immediately the next one, right? Now we have to get rid of this issue, right? The fact that even if you don't have anymore this red arrow here, you still can have interesting crosstalks that happen, right, between circuits just because they are sharing the same resources, okay? So, how do we go about this? You have the issue of having many clients, right, attached to the power distribution line, and you can know that imbalances among the loads. Thankfully, they're not that frequent, but like it happened in New York, right, in few years ago, right, it can quite happen that an imbalance creates a shutdown of the network, right? In the case of the cell, the worst case is that the cell dies, right? The best case, somebody is left without current for some time, and then it comes back, right? And in our case, will be that the cell, right, is starting to grow slowly, right, and producing less output. But in any case, so that's not a new problem. So another very correlated problem is what happens in water distribution lines, right? In water distribution lines, you have, right, essentially, you can have water towers depending whether you have a hilly landscape or not, but then you have water distributed, like in houses, for example, and you probably have experienced that in your same home, if somebody is turning hot water on downstairs and you're having a shower, right? Sometimes damn, is somebody using the hot water down there? Sometimes yes, right? So these are all known problems. So therefore, when we started looking at this issue, we thought, okay, can we leverage, right, any solution that is in this field to our case? And at least we came to the conclusion, unfortunately not, okay? Because the physics is too different, okay? The physics is too different. Here you talk about, right, phasors, right, and you go in the frequency domain description, right? Here we talk about mass flows. It's probably closer, but very different still. So therefore, what we went back to basics, okay, to the central dogma of molecular biology, where you have your DNA takes resource 1, which is RNA polymerase, resource 2 is ribosome, you get the protein, and then the concept is what happens when this is part of a network with many genes, okay? So this is going to be, right, the system where it has an input U, right, and output protein P, okay? And you can have it as part of a bigger network. So there was a quite big effort in writing biophysical models that can have very small dimensionality, okay? Because ultimately, a model that for us is useful for design is simple, okay? So I'll spare you the math behind it, but what we found from the biophysical model is that we can predict the effective interactions that in any circuit happen because you have resource competition, okay? So let's start with the first one. So if you have gene PJ activating another gene, it becomes actually a repressor for any other gene. Why is that? Because when this gene is activating, this gene is demanding resources, right? So if it's not directly activating any other gene, this gets resource depletion, so it goes down, okay? So that's how this hidden interaction comes into picture. You can also have the reverse, okay? If this gene is repressing another gene, it's freeing up resources from this, okay? So when you increase this node, you are actually activating, right, any other node that is not directly connected to it, okay? We call this lateral repletion and lateral activation. And then you can have very interesting things by which the direct connectivity is essentially not changed. So resource competition makes direct connectivity weaker, but the sign is kept, okay? And then you can have situations where, okay, so these properties are parameter independent. So depends only on the structure of the network, so that's why the approach is useful. There are other situations in where the signs, okay, depend on the parameters, and these you have to look at case by case. But in any case, so these are some initial experiments that we did, okay? So you can have a gene which is activating a red gene. We want to test whether a green gene that should always be on is affected. So we put the circuit on a plasmid, okay, with 2 fluorescent proteins, so GFP that you can see, right, RFP that also you can see and you can induce, right, with an input, which is called AHL, it's a small signaling molecule. Now, of course, we had a bunch of control experiments where we essentially removed parts of the system or substituted them to make sure that it's really the process of activation that is the cause, right, of these interactions. And so what you can see here is that in a plot, when you increase the input to this red, right, node, essentially the green node, which is always on, ideally, actually goes down, okay? And this interaction can be up to 70 or 80%. So it's definitely not something you can neglect, okay? So, and this is what actually people, when you design a big circuit, these are interactions that happen all the time, and that's why people go through several iterations of retuning or redesigning the circuit once you have all the pieces there, right, to get rid of these interactions. But the question is, can we create a model-based framework, right, to mitigate these interactions so you don't need to do this iteration all the time? So, essentially, then what you can do, right, you can create simple biophysical models that predict something very interesting. It doesn't matter what circuit you're building, any pair of protein concentration is constrained on a line, okay? Simple. So, if you're producing protein P1 and protein P2, it can be disconnected, it can be actually connected. They are bound to be on a line. If you increase protein P1, protein P2 must decrease, and the decrease is linear, okay? So that's very similar to the concept of isocost in economics, right? Because these proteins are actually trading for resources, and each protein has its price that you can tune with biological parameters. What are the biological parameters you can tune? The circuit's copy number of the gene, and the ribosome binding site strength, for which we have library, right, of different strands, right? We can probably have 50 to 60 different strands, okay? So with this, right, if you tune these prices, you can essentially change the slope, okay? And in fact, we did a bunch of experiments in which you were tuning the copy number, the ribosome binding sites, and now you can see that you can essentially tune the slope, right, of these genes. of these isocosts. So then the next question is, how can we use this framework, okay, to actually fix broken circuits, okay? So here is what happens now, going back to the cascade, right? Here is the expected behavior of the cascade. If you put these hidden interactions into picture, the cascade now becomes what we call an incoherent feed-forward loop, where you have a positive activation, which are the intended regulatory interactions, and now this negative interaction, which comes for a source competition. Well, these are motifs that are studied since, 2000s, probably even earlier. They happen in nature all the time, and they have biphasic behavior. They can have decreasing behavior, they can have increasing behavior, and here is essentially the parameter space that you can build for these systems. Turns out that our biophysical model predicts that there is one parameter that depends again on DNA copy number, ribosome binding site strength, that predict how you move among the different behaviors of the system. These parameters are called resource demand coefficients, and they quantify how much resources each node is demanding for its production. Okay? Therefore, what you can see here is that as these resource demand coefficients increase, you can move from the case where you had your broken cascade, which is completely decreasing, right? The opposite case, to the case where it's the ideal one, in which the interaction is really weak, J is really small, and you have your expected behavior. Therefore, if you want to fix the cascade, you have just to keep this parameter space in mind and move your system through this space by changing ribosome binding sites and copy numbers. Okay? So here is the experimental setup. You have your parameter space. These are the behaviors seen in each one of these regions. We started from the broken cascade. Okay? But now let's explore this parameter space. If you go this way, you really break the cascade. Okay? And that's done by increasing the copy number. Well, it's even worse, right? Even more decreasing. But then let's try to go back toward the good region, okay? Let's therefore increase the ribosome binding site, and well, almost there, okay? But then let's try to go back all the way in this white region where the good behavior is. That's the minimal resource demand coefficient that you can get. And yeah, okay. So the cascade was fixed, okay? So therefore, with this, we now have at least in place a tool that by changing ribosome binding site strength and copy numbers, which are really easy to change in synthetic circuits, right? You can change this resource demand coefficient to move you through the parameter space. Okay, so that's essentially the idea. Okay, so any questions so far? No questions? So let me therefore just give you a snapshot of what we are doing right now, but I won't go into too much details here. So this is a situation where how many ways do you have to fix your circuit? One way is to tune the parameters of all the parts, as I've shown you, right, by changing this resource demand coefficient. The other one, which is probably better for modular design, is design each circuit component such that it is careless of its context, right? So that it's robust, right, to the way the context changes, okay? So essentially, you want to go from a situation where you have your regulatory interactions with hidden interaction to a case that the system behaves like this, okay? And the way you can do that is by adding negative feedback mechanisms at each node in a decentralized fashion, okay? So each node is going to have its feedback mechanism that makes that node robust, okay? Essentially to the red arrows that you can view as disturbances, okay? Yet again, we have the concept of negative feedback being used to make input-output response robust to perturbations. And that's your system formulation. Once you have your system formulation in these terms, essentially the problem is half solved, okay? So you can essentially view each node as a system with inputs and outputs, which are black, right? These are contributed to the intended regulatory interactions. And then you have disturbances, which are essentially the fluctuations imparted on the resources concentration by all the other nodes, okay? So the problem is that you want to design a feedback mechanism around this to make the V to Y response independent of these disturbances. So, you can mathematically formulate the problem. I will just show you the formulation. I won't go through the proofs, okay, that we also do, by the way. But essentially, you can compare two systems, a system which is not in the network, that doesn't have resource perturbation on top, to a system that has resource perturbations on top, and ask that the two outputs are close, okay? So how do you do that? Well, you want to design a feedback mechanism in the system such that the response of B to Y is independent of the disturbance, and you make sure that this loop doesn't create instabilities in your system, okay? So that's a network stability problem. So that requires a little bit more mathematical machinery that I won't go into, so I will just show you how for a single node we solve, right, this disturbance attenuation problem. And here it comes the, I think, probably most exciting discovery that we have done in the past probably 2 years in collaboration with some folks that do actually study mammalian synthetic biology, which is this concept that you can engineer actually an integral control through phosphorylation, okay? So integral control is used in engineering, right, to achieve at least with constant disturbances and constant input, perfect adaptation, right? Is essentially the mechanism roughly by which your automatic cruise control work, right? So you have a speed set, right? You have maybe a slope perturbation. Doesn't matter what the slope is, your vehicle is going to track that speed, okay? So integral control is an integral part of that, okay? And it turns out we can do that in biology as well. And it turns out that these interesting, again, phosphorylation cycles, if you have them work in a situation where the enzymatic reactions are saturating, you can actually have almost perfect integrators, okay? So what happens is that this is essentially what biology tells us. We use toy models, okay? So this is a very toy model that you can write down that is an ordinary differential equation that looks at the rate of change, essentially, of the output of your system. These are standard textbook models for phosphorylation systems, okay? So take any textbook that's standard. What you can see here is that if you essentially make sure that these constants, which are called Michaelis Menten constants, are really small, okay, you can reduce this differential equation to a pure integrator, okay? You see that x star is the pure integral of the error between the input and the output, okay? And of course, if the system is stable, right, at steady state, you should expect that independent of whatever disturbance is happening in your feedback loop, right, your output should track the input no matter of what disturbances you have. So essentially, this is what you expect. The open loop system, where you do not have this controller, you have when you add disturbance, meaning fluctuation of resources, a difference in the response. With the feedback, the difference should be quenched, okay? So, this is essentially the system that we built in the past 2 years. You have, so this was built in mammalian cells, and we essentially took as our integral controller 2-component signaling system that we extracted from E. coli, for which we could mutate the kinase and the phosphatase, so they could be exclusively kinase and exclusively phosphatase. For those of you that you know a little bit about these systems, most kinase and phosphatase are promiscuous. Some of them are both activities, right? So we had to do some mutation there to make sure they were specific. But essentially, we have a disturbance generator is a system that is activating a gene and sequestering resources from your output gene, okay? That's showing the disturbance is working, okay? Here is the behavior of your output protein, essentially with this loop broken, okay? So that's the open loop system. When we put back this loop, we go to this, okay? So essentially you have that the response input-output is essentially independent of various level of disturbances applied, okay? And by the way, the response input-output is almost linear because that's what actually an integral does, right? So the output is proportional to the input, okay? So that's the bottom line from here. So we now are building various library of the system. We already have 2 in hand, okay? And since bacteria offer a large number of orthogonal, right, to signaling component systems, right, you can actually build a pretty large library with that. So, anyways, So that's the interim summary. I want to spend the last 5 minutes to tell you about something really exciting, okay, that we got into about 2 years ago, which is about really this notion of being able to control self-fate by engineering feedback controllers in synthetic circuits, okay, that now interact with endogenous cellular machinery, okay? So here a little bit of background. I've already shown you this picture. You have pluripotent stem cell up here. You have differentiated cell. Differential potential goes this way. You can have transdifferentiation. And the Nobel Prize in 2013 showed that you can overexpress some key factors, which are called OCT4, SOX2, CAL4, and c-MYC. It's called the OSKM. Which can take a fibroblast, is like a skin cell, which has very low values of this, while a pluripotent stem cell has very high values of this. So what they did, they cloned, right, this OSKM into the virus. They infected fibroblast cells, right? The virus inserts the OSKM in the chromosome, right, of the cell. The cell starts expressing it, and wow, you can go back here, okay? So OSKM, it's high, you can get a pluripotent stem cell. Okay? That was fascinating, right? That destroyed actually the belief at the point that this process cannot be reversed. Okay? So, what's the problem with this? So today still the efficiency, as I was saying, is very low, right? In a recent review, Yamanaka said that they tried the first clinical trial with these, where you actually take skin cells from a sick patient, you convert them to pluripotent, and you differentiate them back, right, to, for example, a liver cell or some other cell type that the patient is needing. But instead, the process takes too long, it's too expensive, and it's very efficient, so it's not applicable really yet to clinics, okay? So, therefore, we actually asked, why, first of all, the intriguing question is, why is so inefficient, okay? Of course, that's a big question, it's a holy grail, right, of the field, right, many people are after. But the second question we asked, can we get rid, right, of this problem to some extent using feedback, right? So here is where we look back at the G regulatory network that actually control these transitions, okay? So I won't go too much into the details, but you can view the problem of controlling self-fate as the problem of moving the transcription factor concentration, which determines the state of the cell, right? For example, high values pluripotent, low value skin cell, right? As a phase plane system where you have multiple stable attractors, right? Each one of them could be a cell type, okay? Could have pluripotent here, could have skin cell here, I could have primitive endoderm here, right? Then the idea is that you apply an input, it's plus u, it's an overexpression, so you add these transcription factors in the system, and now you change your system such that if you essentially are trying to get to S0, you can actually get close to it independently of where you start, right? So ideally you have one unique attractor really close to where you want. So when you remove the input, right, you go back to it, okay? So in mathematical terms, right, this is essentially formalizing a little bit this concept of reprogramming self-fate, okay? So once you view it in these mathematical terms, then you can ask, is a certain network reprogrammable, right, to the state that you want, right? And what you have to be able to say for being reprogrammable to a target state as 0 is that there is an input, like in this case, such that you can move any state close enough, right, to the state that you want. Okay, so that when you remove the input, you're essentially in the basing of attraction of the target state, and you're going to converge there. Okay, turns out that if you take a closer look to the pluripotency network, the pluripotency network is one where you have all positive interactions. So that's a class of network that in dynamical systems is called cooperative network. Okay, it's very well studied. And for this network, you can actually demonstrate that if you're trying to reach an intermediate state, okay, so a state that has intermediate values of some of the transcription factor concentration, you cannot do that with pure overexpression. Okay, and it turns out, look at here, the pluripotent state is actually having OCT4 concentration that is intermediate, okay? So if you overshoot it, you go somewhere else. It's not pluripotent. This means that stringent control is required, right, to get you there. So the idea is essentially this. If you view, right, your pluripotent state in the middle, you have differentiated cells on the left, on the right, you have a valley. These 3 points are all stable, right? So if you put a ball here, it stays there. With pure overexpression, you essentially risk to overshoot it, okay? So that's the problem. So what do? We use feedback, okay? So your input u now, instead of being a constant, right, overexpression, it's an expression level that adjusts itself based on the difference between where you are and where you want to go, okay? If you go back and look at the math, of course you can prove beautiful things. I won't go to that, but in a picture, this is what it means, okay? So pure expression can get you to overshoot. Pure degradation can get you to overshoot the opposite way. But if you put both of them together, it gives you a feedback mechanism. You can create a system that has a unique attractor, hopefully centered where you want, okay? So that's what high gain feedback does for you, okay? So that said, we are building a genetic circuit controller that actually is able to have high gain feedback to control this transcription factors concentration. The way we do that is essentially having an amplification here through overexpression, and then you need an enhanced degradation that we can get with small interfering RNA or microRNAs, okay? So that's a very powerful way to get this huge negative feedback, okay? So if you do that, of course, you can, yet again, what we do, create a beautiful biophysical model for which you can prove properties, but then ultimately, if you take a huge computational model, stochastic, and etc., what would predict is that with the controller on, you can essentially take all your cells, so each trajectory is a different cell, all your cell in about 10 days, right, to the value of your oct SKM target, okay? Then, of course, when you remove the controller, it's the cell endogenous circuitry that keeps you there because you move the system in its regional attraction, okay? So anyways, we are building this system. We have already built the OCT4 controller. It seems to work. Of course, to reprogram the cell, right, we will need to build the other 3 controllers, right, for SKM. Anyway, so that's a summary. I want to point out that if you want to learn a little bit more about the fundamentals, there is a textbook that you can read about most of them, okay? And then these are really all the people that did the work. I typically get credit, but these are really the key people in the talk. My student, or the first student is this. Yili Hyun, who's going to graduate next year. Now you can see here was half happy, now is really happy, okay? And that's why he's graduating next year. I want to thank in particular Eduardo, that has been really key in setting up the first wave of this all foundational theory, and with whom I always have a really intriguing discussion, including today. Okay, so that's the summary. Thanks.